a bit at the beginning. So just to review those things that you should know going into that, that's what we're doing right now. And some of these things will be coming up, you know, as we talk about those things. So we were talking about how in, let's see if I can get this to go forward or backwards. In the dendrites and cell body, that blue zone in the picture there, you have, if a nerve cell, the neuron is stimulated appropriately, it can generate what are called graded potentials. Now remember that we talked about last time, the resting membrane potential in a neuron is around 70 or negative 70 millivolts. And as I mentioned, that means that if you're looking at the plasma membrane of the neuron, the inside of the membrane is more negative than the outside. If you measure with a probe, it measures negative 70 millivolts. It's you know, partly because of the, or mainly because of the sodium potassium ATPase pump. You have an uneven number of positive charges, and that is perceived by the voltmeter as a negative, right? So you have more negative in here, more positive on the outside. So that's called the resting membrane potential, meaning the neuron is at rest. If you stimulate the dendrites or cell body appropriately, then you can generate graded potentials. These are a change in the resting membrane potential. They're not nerve impulses, but they are electrical signals that can generate a nerve impulse. And these graded potentials can either be hyperpolarizing or depolarizing. Depolarizing meaning, you know, if this is polarized, if it's polar because there's a positive and negative side to the membrane. Depolarizing means it's becoming less polarized. It's, you know, more positive on the inside than it was before. It's going towards zero, so it's depolarizing, becoming less polarized. Hyperpolarizing is becoming more polarized, more negative on the inside. And the way that happens is when the neuron is being stimulated in that blue region there, there's the opening of gates, gated channels. These are protein gates. They have a gate on them. When the gates close, nothing's flowing through. When the gates open, something can flow through the membrane, flow through the protein channel. Keep in mind that ions, sodium ions, calcium ions, chloride ions, are all charged ions. Because they're charged, they can't penetrate the phospholipid bilayer. Because they're strongly charged, positive and negative charges or strong charges, they can't penetrate those fatty acids in the phospholipid bilayer. So unless there's a protein channel, they're not going to get through the membrane, okay? So there's a couple ways you can stimulate a neuron. If it's a sensory neuron, specifically one that responds to either touch, pressure, or vibration, and that would also include sound waves. So some of the, you know, receptors in your skin, as well as the receptors in your inner ear that detect sound waves. They are going to be, when they're stimulated by the appropriate stimulus, that is going to open up the gate. And when the gate opens, in flows positive ions. And when positive ions like sodium or calcium flow in from the outside to the inside of the cell, the yellow is the inside, the cytosol, the outside is the extracellular fluid, the blue is. So let's go back to this picture. So if you've got positive ions flowing in, it's going to become depolarized. It's going to become more positive on the inside. And but there's another way that cells can be stimulated. This neuron is multipolar, so in this picture, it's not a sensory neuron. Multipolar neurons are either motor or interneurons. And those neurons aren't stimulated by touch or things like that. They are stimulated by neurotransmitters, okay? So there's some other neuron in the pathway that's stimulating this cell at a synapse with the dendrites or the cell body. And so for those kinds of neurons, for motor neurons and interneurons, they're going to have ligand-gated channels in this membrane. And those ligand-gated channels are opened by neurotransmitter. If I can get this to go. So a ligand, the word ligand means a chemical messenger. So it could be a neurotransmitter, it could be a hormone, it just depends. In this case, it's a neurotransmitter. Excuse me. And there's two kinds of neurotransmitters, excitatory ones or inhibitory ones. Excitatory neurotransmitters, acetylcholine in certain cases is excitatory, in other cases it's inhibitory, but it can be an excitatory neurotransmitter. And so here it is this would be the postsynaptic membrane. This is the membrane of the cell that's being stimulated. And it has these ligand-gated channels for acetylcholine. The acetylcholine is the little red sort of pyramid shaped or triangular shaped things here. And they have a special binding site in two places on that acetylcholine receptor. And if the acetylcholine binds in both places to the receptor, it will open up the channel and inflows sodium and calcium ions. I don't like this picture because it's showing an arrow going in the wrong direction, but it should be showing, there's a reason for that, but ignore it. It should be showing just, flow of sodium and calcium into the cell primarily. That's going to make it more positive. So, the inside of the membrane was originally more negative, now it's becoming more positive. It's not completely positive in reality, it's just getting a bit more positive, so it's depolarizing. So when this happens, you're going to get a wave like this, just like you got with the previous picture. So let's look at a synapse. Here you have a synaptic end bulb at the end of an axon terminal. This is one synapse. Here is the postsynaptic membrane. So this is where those ligand-gated channels are located, in the dendrites and cell body of the neuron, the postsynaptic neuron. So in other words, these are going to be found here in this neuron, okay? And so if the presynaptic neuron is sending nerve impulses or action potentials down to the synaptic end bulbs, that's going to open up calcium channels, voltage-gated calcium channels. These electrical signals are, these nerve impulses have a voltage, so they open this gate, these voltage-gated channels. In comes calcium, and we'll talk a little bit more about this in a minute, but calcium facilitates the release of acetylcholine, or whatever the neurotransmitter is. These synaptic vesicles are sacs, they're membrane-bound sacs, and they contain neurotransmitter of some type. Could be acetylcholine, could be a different one, but it's going to be one type of neurotransmitter. When calcium flows in, it sets into motion a series of events that results in these vesicles traveling to the very tips of the synaptic end bulb, the membrane fusing with the plasma membrane of the synaptic end bulb. So here it is fusing and releasing the neurotransmitter by exocytosis into the fluid-filled synaptic cleft, which is a tiny space between the synaptic end bulb and the postsynaptic membrane, the membrane of that second neuron. The neurotransmitter shown in brown travels across the synaptic cleft, binds to the receptor for that neurotransmitter, and normally you have two receptor sites. So if only one gets bound, if one only binds, then nothing happens. You have to have two. It's like a trigger. Once you have those two acetylcholines attaching to that ligand or ligand-gated channel, that receptor site, then the gate opens and inflows some kind of ion. It could be a calcium ion, it could be a sodium ion, it could be chloride ions. But that flow of ions is going to create a graded potential in this cell. And we'll talk more about what happens to that gated potential, or that graded potential, in just a second. So in the case of that example that I showed you, if it's acetylcholine binding to its receptor, and if this is in the appropriate place, acetylcholine can be inhibitory, but in this case excitatory. If you have a flow of sodium ions through that gated channel, it's going to make it more positive on the inside, which means it's going to be a depolarizing graded potential, and we'll see that that can be more a stimulatory thing, not an inhibitory thing. In the case of other neurotransmitters like glycine, glycine is a neurotransmitter in the brain. It's inhibitory. When it binds to the glycine receptor, it opens up a gated channel that lets in chloride ions. Chloride ions are negative ions. That's going to make it more negative on the inside, which means instead of getting this, you get this. This is when you get a hyperpolarizing graded potential. When you have a flow of chloride ions into a neuron, that's going to create a hyperpolarizing potential, which means it's more negative on the inside. And that's really taking it away from threshold, and we'll talk about threshold in a minute. So this is inhibitory. And so there are inhibitory neurotransmitters, there are excitatory ones, and we'll talk about why they're inhibitory or excitatory in just a minute. But those are the two possibilities there. So that's how you get those graded potentials in a neuron's dendrites or cell body. Again, the top picture is happening in sensory neurons, in the dendrites of certain sensory neurons, which are either unipolar, well, let me think if any of them are bipolar. I think they're all going to be unipolar in this example. But this could be happening in the multipolar neurons, dendrites, and cell body of those motor or interneurons, okay? So, moving on. So these postsynaptic potentials, or I should say graded potentials, we haven't used that term yet, but these graded potentials that are generated in the postsynaptic membrane, which are called postsynaptic potentials, they could generate a nerve impulse in the axon. It depends. If it's a depolarizing graded potential, it could lead to a nerve impulse. But if it's an inhibitory or hyperpolarizing graded potential, it's actually going to inhibit the formation of a nerve impulse. And this is why. When you get an action potential, which is this picture right here shows an action potential. This is what an action potential looks like. It's a nerve impulse. And this is showing the millivolts of an action potential. Action potential rises up to about 30 millivolts there at the top, and then comes back down to resting membrane potential. This happens in the axons. This does not happen in dendrites or cell bodies. It happens first in the axon hillock or in the trigger zone of the axon, and it continues on down the axon all the way down to the synaptic end bulbs. When we talk about a nerve impulse, this is what you're really talking about, is this type of change in voltage. And in order to get a nerve impulse, in order to get an action potential, you're going to have to depolarize the membrane to threshold. Threshold is negative 55 millivolts. So if you just slightly stimulate the neuron, either by slight, stimulation of a mechanically gated channel neuron, or by just a little bit of neurotransmitter that depolarizes the membrane just a little bit, this is subthreshold. That's a subthreshold stimulus. In other words, if you stimulated a pressure receptor or a touch receptor just slightly, and you only got a subthreshold stimulation, you wouldn't feel it because there wouldn't be enough stimulation to generate any action potentials that send a signal to your brain and tell you you're being touched or there's pressure. So this is subthreshold. Nothing's happening. If you, however, apply enough stimulation to get the membrane to threshold, which is negative 55 millivolts, then automatically you'll get an action potential. That'll automatically happen. So you have to depolarize the dendrites and cell body to threshold. And then as long as it's at threshold at the axon hillock or at the trigger zone of the axon, then an action potential will be generated and then propagated down the axon. So that's one of the things that you have to keep in mind is you have to hit threshold. You have to get enough stimulation. And we'll come back to this in just a minute. But an action potential is generated not by ligand gated or mechanically gated channels, but instead by what we call voltage gated channels. Voltage gated channels are opened by a certain amount of voltage in the membrane. Anything less than negative 55 is not the proper voltage to open these gates. So anything, you know, under negative 55, if it's at resting membrane potential or subthreshold, these gates remain closed. The gates that we're talking about, these are gates located in the axon membrane, in the axon hillock or axon trigger zone, and all the way down the axons to the, you know, synaptic end bulbs. You have these voltage gated channels, and they are sodium gates and potassium gates. Now this book of yours gets into these, there's actually 2 gates in the sodium channel. I'm not gonna worry about those 2 gates. I just talk about gates in general. So if any of these gates are open, then, you know, the gate, it's gonna allow sodium through. So, or I should say both gates are open, sodium can flow through. So I don't worry about which gate is which. I don't get into that kind of detail. But if the gates are closed, like they are here, the gates close in the potassium channel and in the sodium channel. Sodium channel is in green, potassium channel is in yellow. And one thing you want to keep in mind is there is a concentration gradient of sodium and potassium. Remember, we got the sodium-potassium ATPase pump. I talked about before, it pumps out, remember, 3 sodiums and then pumps in 2 potassiums. So you have not only a charge difference, but what this, you know, so this is part of why you have a resting membrane potential of negative 70, but this also creates a concentration gradient of sodium and potassium. That means you have more sodium out here, and you have more potassium inside the cell. In other words, relative to, you know, more sodium, I should say more sodium and less sodium, because that's all that really matters there, and then more potassium here, less potassium out here. So higher concentration of potassium inside the cell, higher concentration of sodium outside the cell. You got a concentration gradient, which means you can have diffusion if those gates open. So the sodium, you know, sodium's going to diffuse which way? If you open the gate, which way would sodium diffuse? In or out? Which way would it go?
Go in.
It would go in. Yeah, if you have more sodium, sodium will naturally, if it can diffuse through a channel, it'll naturally diffuse in. Potassium, on the other hand, would diffuse out, wouldn't it? Okay, that's the natural. So if you know the concentration gradient, you kind of figure out which direction it's going to flow, but you have to know the gradient. But that goes back to that sodium-potassium pump, which is a protein pump that keeps all these gradients going. So at rest, you're at negative 70 millivolts. There's nothing happening. The gates are all closed. But if you stimulate the neuron, and you stimulate it so you get a depolarizing graded potential. If the depolarizing graded potential reaches negative 55, which is threshold, they don't show it on here, but if it reaches negative 55, that's going to open the sodium gates, only the sodium gates. Potassium gates still closed. So notice that sodium is flowing in, and when sodium flows in, I'm going to show you this next picture here. So here is negative 70 at rest, you stimulate it to threshold, you depolarize it to threshold, you're at negative 50. Now sodium is flowing in, and so you can see it's rising. It's becoming more positive on the inside, eventually reaches a point where it hits 0 millivolts, continues to go up to about 30 millivolts. At 30 millivolts, the sodium gates close, and the potassium gates open. So let's go back to this picture. So sodium is flowing in, and the graph is rising, the line is rising. It's becoming less polarized, it becomes unpolarized, and then it has a reverse in polarity. It actually becomes more positive on the inside than the outside. At 30, there's actually more positive ions on the inside instead of more positive ions on the outside. So you reverse the polarity. At this point, at 30 millivolts, the sodium gate closes, and the potassium gate opens. Now potassium flows out, and potassium, as it flows out, As potassium flows out, the line goes back down because now you've got those positive charges going back out again, making it more negative on the inside. So eventually it's going to go back down to its normal resting membrane potential. It goes back down to -70. Sometimes it goes a little bit lower, but it gets kind of reestablished by the sodium potassium pump and it's back to normal. So it's back down to resting membrane potential. That's as potassium is flowing out and taking those positive charges with it. It's going back down to its negative millivolts. And so this happens until you hit, about -70 again, and then the potassium gate closes. Now you're back to having both gates closed. So you have first the sodium gates opening, sodium flowing in, then that gate closes, potassium gate opens, potassium flows out, and you're back down to -70. That's one nerve impulse, one action potential. And this is happening in the axon. It begins at the trigger zone, which in this kind of neuron is the axon hillock, a little bump at the beginning of the axon. If you're if you're stimulating these dendrites and you get it to threshold, then if it's at threshold at the trigger zone, because these graded potentials can flow down the membrane, when they hit the trigger zone, if it's at -55, then you automatically get an action potential, and that action potential will continue or get propagated down the membrane. It just gets propagated. One action potential leads to another, which leads to another, and it propagates down the membrane. Notice too that if you look at the previous picture, you can really see this. We're talking time in milliseconds. This is like the time, like electric currents. They're moving at a rate of milliseconds. So you're talking very fast. These things, the sodium and potassium gates open and close within milliseconds, not within a second. So it happens in less than a blink of an eye. So these are very, very fast processes that are happening. And then that action potential being propagated down the membrane, that is happening very quickly as well. So these impulses travel very fast, maybe not as fast as electricity, but pretty fast. So this goes into a lot of more detail than I do, but it does show the different phases. Here's membrane potential in millivolts, here's time in milliseconds, here's stimulation. If you get it to threshold, then you get one action potential. Depolarizing phase corresponds to sodium gates opening and sodium flowing in. The repolarizing phase corresponds with potassium gates opening and potassium flowing out. Meanwhile, the sodium gates are closed at this point, and then you get back down to resting membrane potential. Any questions about that? So again, these action potentials propagate each other because when you depolarize the membrane here, that functions to bring the next section of membrane to threshold, and then you get another action potential. So one action potential will create action potentials further down. When you're depolarizing the membrane, that's going to cause an action potential to be generated right next door, and it's just going to propagate all the way down the membrane. Okay, now action potentials, unlike depolarizing graded potentials, which vary in strength, the action potential does not. And by the way, I want to point out, if you're hyperpolarizing, notice you're hyperpolarizing, taking away from threshold. That means you're inhibiting action potentials. So that depolarizing can stimulate, but inhibitory, or hyperpolarizing are inhibitory. They prevent action potentials. But anyway, when you get an action potential, that's the size of it. It goes up to 30 millivolts. Even with a super threshold, in other words, above threshold stimulus, in other words, you're stimulating a whole lot, you don't get bigger action potentials, you get more of them. So the more you stimulate the neuron, you're not getting big action potentials, you're getting the same size action potentials, just more of them being propagated down that axon. And so there's a picture here of an example of that in an experiment where if you were to apply a different size weights to the skin, to an area of skin, if there's only about a 1 gram weight there at the top, and this is a measurement of the action potentials here, it's a graphical representation, each little peak is one action potential. So 1 gram just barely stimulates that, pressure receptor in your skin, those pressure receptors in your skin, and barely, generates enough stimulus that sometimes gets an action potential, but, you can see they're just scattered action potentials, just barely enough that sometimes you hit threshold, but just barely, so you're just barely stimulating the mechanically gated or the sensory receptors, and just barely opening some mechanically gated channels, and every once in a while you get enough sodium in that it depolarizes it to threshold. So you can see, I need a pointer in here or something, but you can see occasionally you do get an action potential. So it's gonna be a very weak stimulus. It's gonna be perceived as a very slight pressure, probably barely even felt pressure on your skin. The more weight you apply though, the more you're stimulating those neurons, those sensory neurons, the more mechanically gated channels you open, the more action potentials are being generated and then propagated down the axon. So with each added weight, you can see more and more action potentials. This is being propagated then down the membrane, so you're getting, boom, 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 tons of action potentials going, one after another, traveling down the axon. And then the axon releases neurotransmitters, stimulates another neuron, an interneuron in the pathway, and eventually this information gets to the brain. The brain interprets this as, it uses this as data, and so it's going to interpret that as very light pressure, a little bit more pressure, a little bit more, so that, the difference that you're sensing, that you're perceiving in your brain, is based on how many action potentials are being sent to the brain at that moment. So this is how your brain is determining the difference between a heavy weight and a light weight. Does that make sense? So this is data for your brain. Your brain is interpreting that data, and then taking that data and processing it like a computer processor and saying, okay, you're feeling a lot of pressure versus just a little bit of pressure on your skin. Or maybe no pressure at all. Probably 1 gram, you might feel it, might not, If you didn't see it was on your skin, you might not even notice it. Okay, and we're gonna be talking more about that later when we get to the sensory system, but I just want to remind you that, these synapses occur usually, when you talk about synapses, between like a sensory neuron and an interneuron, or between an interneuron and a motor neuron, or between interneurons in the brain, things like that. You have a presynaptic neuron that's sending the action potential, sending the impulses. You've got a postsynaptic membrane or postsynaptic neuron that's receiving stimulation from those presynaptic neurons. And these are the axon terminals of the presynaptic neurons, shown in blue, green, and red. And then the little synaptic end bulbs that release the neurotransmitter that we talked about. This is the dendrite and cell body of a multipolar neuron, either motor neuron or interneuron. Most synapses are either axon to dendrite. It's called axodendritic, but don't worry about that. So you have receptors, which I'm assuming are the bumps on the dendrite here, but you have receptors for that particular neurotransmitter that releases. Each neuron releases a certain type of neurotransmitter. It doesn't release multiple kinds, it releases one kind. So it's going to have receptors for that neurotransmitter. This one is showing another synapse between the axon of the presynaptic neuron and the cell body. So these are the two most common types. This is where graded potentials are generated. Those postsynaptic potentials or graded potentials, excited to, or depolarizing or hyperpolarizing, are being generated in the dendrites, and then those graded potentials will travel to the axon's trigger zone, which here would be the axon hillock. There are, more rarely, but there are axon to axon synapses where the axon, presynaptic one, directly stimulates the trigger zone, but that's not very common. It's usually this. So usually what we're talking about is this kind of case, and we don't really focus much on that. So, but I am pointing out that you do have that in some cases. So keep in mind that here you're going to have those graded potentials, either depolarizing or hyperpolarizing graded potentials. Here's where you begin the action potential. It's then propagated down the axon. remember that the myelin sheath, if there's a myelin sheath, the propagation happens at these places where there's no myelin sheath. These are called nodes of Ranvier. Nodes of Ranvier. You can see that in the picture of the neuron. And so the action potential here will propagate an action potential at this node, which propagates an action potential at this node. And so they often say that the action potential jumps from node to node. It's not really jumping, but there is propagation. You know, that flow of sodium ions into this part of the axon will depolarize this membrane to threshold. And then when its sodium ions flow in, that depolarizes this axon, this node of Ranvier to threshold. And so this is much faster. When you have myelinated axons, you have much faster propagation versus unmyelinated axons where the propagation is continuous and just flows down the entire axon. So myelin sheaths like this help in faster action potential propagation. Any questions about this picture or anything else that we've talked about so far? Okay. This is another picture that shows the synaptic end bulb and the synapse. In other words, we're looking at a highlighted view of one of these synapses here. This is the synaptic end bulb. This is the postsynaptic membrane. What I want to point out is that when action potentials arrive at the axon terminal and at these synaptic end bulbs, again, it opens voltage-gated channels. These ones are calcium channels. They're opened by the voltage of the action potential, okay? So when that voltage arrives, when that action potential arrives, this gate opens, inflows calcium, and what calcium does is it activates various proteins. Oftentimes, as we'll see, the way things work in a cell is that one thing leads to another. And what often happens is that you activate proteins that act as internal chemical signals that activate other proteins, which activate other proteins and set things into motion. That's how you get things to happen in physiology. In this case, calcium activates a protein called calmodulin. Calmodulin activates inactive protein kinases, which are very common enzymes that activate other processes. So here's inactive. Calmodulin activates protein kinase, which in turn activates synapsin proteins by phosphorylating them. You don't have to memorize all this. I don't expect you to know this, but eventually we will talk some about these kinds of systems. But this activates this, which activates this. The synapsin proteins are thought to hold on, in a sense, hold on to those synaptic vesicles, and when they're activated, they release them. So they release them, freeing up those synaptic vesicles so that they can migrate to the end of the synapse here, to the end of the synaptic vesicle, or synaptic end bulb. So synapsin, by activating it, it allows the synaptic vesicles to flow to the, migrate to the very tips of the synaptic end bulb, and then those vesicles, because they're membrane-bound vesicles, they fuse with the plasma membrane, releasing the neurotransmitter by exocytosis, and then that neurotransmitter flows, diffuses across and binds to the ligand-gated channels, the receptors. I mean, those neurotransmitter receptors are also ligand-gated channels, and then, from there, just like we talked about before, when that neurotransmitter is binds to its receptor, it opens up the gated channel and inflows either positive ions or negative ions. If it's an excitatory neurotransmitter, it's letting in positive ions like calcium or sodium. If it's an inhibitory neurotransmitter, it's letting in chloride ions, which are negative, making it more negative on the inside. But what I wanted to show you was, how this leads to that is the activation of proteins that then allow synaptic vesicles to migrate to the end of the vesicle. So like I said, some the excitatory neurotransmitters really open up gates that allow calcium or sodium in. Inhibitory allows chloride ions to flow into the cell. And so we talked about, hyperpolarizing and depolarizing graded potentials. And these are called, when you're looking at a synapse like this, We call those postsynaptic potentials because they're the postsynaptic membrane. And more specifically, we would call, if I can get it to go, okay, so this would generate depolarizing graded potentials, like this one on the bottom. Unfortunately, the pictures don't go together. This goes with the bottom graph. I wish they were flipped over, but anyway, depolarizing here, which we would call an excitatory postsynaptic potential, okay? It's just being a more specific name, specific to the postsynaptic membrane. Excitatory postsynaptic potentials, or EPSPs, can depolarize the membrane potentially to threshold and generate an action potential. On the other hand, glycine opens up chloride ion channels, right? Makes it more negative on the inside, generating this graph, the hyperpolarizing graded potential, which we would call in this case an inhibitory postsynaptic potential, meaning it inhibits. When you ever hear inhibit, it means either slows down or stops or prevents something from happening. In this case, inhibiting nerve impulses. So that means that your nervous system not only can stimulate things to happen, it can prevent things from happening, okay? That's important because, you know, certain things you want to have happen, other things you don't want to have happen at the same time. So, I'm good to go here. Okay, so, you know, whatever happens here, whether it's excitatory or inhibitory, is going to, you know, is then going to determine whether you're going to have a nerve impulse, whether you're going to get an action potential. Now, one stimulation may be a subthreshold stimulation. Let's say that neuron here, neuron 1, the blue neurons are the presynaptic neurons, the red neuron is the postsynaptic neuron, and the red graph means the membrane potentials generated in the postsynaptic neuron. So in the dendrite and in the dendrites of that postsynaptic neuron, if neuron number 1 up there, the blue neuron number 1, releases some neurotransmitter, it might open up enough sodium channels in that postsynaptic membrane up there to get a little blip, to get a subthreshold stimulation. And then let's say a few seconds or a few milliseconds later, neuron number 2 releases some neurotransmitter, some excitatory neurotransmitter that opens up ligand-gated channels and positive ions flow in, but you only get a little blip. Again, it's subthreshold. So individually, you're not getting enough stimulation of that postsynaptic cell to get an action potential. However, if both 1 and 2, and here it says many, because you could have more than 2, you could have several neurons doing this, but if, let's just say 1 and 2 simultaneously, at both at the same time, fire, and by firing, I mean they release neurotransmitter at the same time, kind of like firing a gun, but they're firing out neurotransmitters, and those neurotransmitters open enough gates, you know, you could summate or add up. In other words, the combined effects, you're adding up depolarizing graded potential, so you summate the threshold. Now you get an action potential. So sometimes it takes the effect of more than one neuron. That's called spatial summation. Spatial summation means you're getting stimulation from more than one neuron at the same time, and that's summating to threshold so you get an action potential. So sometimes it takes the stimulation of more than one presynaptic neuron to get an action potential. Another possibility is here you only have one presynaptic neuron and one postsynaptic neuron. Presynaptic neuron releases neurotransmitter once, doesn't do anything, does it again, but not enough, you know, subthreshold stimulation, not enough to get an action potential. But let's say this presynaptic neuron releases neurotransmitter once, and then again, and then again, and again, meaning you got action potentials propagating down that presynaptic neuron, stimulating more and more release of neurotransmitter, boom, 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 and it summates to threshold. That's called temporal, meaning over time. You're getting stimulation one after another, not all at once by 2 or more neurons, but one after another. That's called temporal summation. So that's another way you can summate and get to threshold. And in reality, you can have both happening at the same time. You can have several neurons releasing neurotransmitter either in waves, boom, 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 or all at once. So they could be doing both spatial and temporal summation all at the same time, or at different times. But in this case, they're also showing that you've got some inhibitory neurons releasing inhibitory neurotransmitters generating IPSPs, inhibitory postsynaptic potentials. Some of these neurons are releasing excitatory neurotransmitters generating EPSPs at the same time. And whether you summate to threshold or not depends on how these all add up. If you've got negative hyperpolarizing potentials at the same time as you've got depolarizing potentials, however they summate, if you get it to threshold, you get an action potential at the trigger zone. If not, nothing happens. So if you get maybe more excitatory neurotransmitters released than inhibitory, you're more likely to get a nerve impulse. If you have more inhibitory neurotransmitters released than excitatory, then you very well won't have a nerve impulse generated. So there's a lot, it's more complex than just simple, one thing or another. You can have a lot of stuff going on at the same time. Any questions about that? Keep in mind that each of those neurons, those presynaptic neurons, is receiving signals from the brain, and some of those might be excitatory signals saying, do this. The other might be more inhibitory saying, don't do this. It could be, for example, whether or not you speed up the heart rate or not, and so the excitatory neurotransmitters might be saying, yeah, speed the heart rate up. The inhibitory ones are saying, no, slow it down. Whether you speed up or slow down the heart depends on which one is having the bigger effect. Is it the excitatory ones or the inhibitory ones? Which one is, they're more of at that instant? Any questions about that? Okay, so I just want to review very quickly about some structures, the spinal cord and brain. Again, the spinal cord, here in this picture, running down the vertebral canal. The nerves, these numbers refer to the spinal nerves, which corresponds with the vertebrae. And except you have one extra cervical nerve, then you have cervical vertebrae. You have 8 cervical nerves because the first one comes out above C1. But otherwise, the nerves pretty much correspond with the number of vertebrae that you have. You even have some nerves coming out of the sacrum and one nerve pair coming out of the coccyx. And that's what a nerve looks like. Nerves are basically bundles of axons, and they're held together by connective tissue. You have all these little bundles called fascicles, kind of like you have in muscle. You have fascicles of muscle fibers. Here you have fascicles of axons. Some of these axons are going to be myelinated. Some aren't. Some of these axons are motor neurons. Some of them are sensory neurons. Some of them are autonomic. Some are somatic. It's a mixture. They're not, divided up in any way. They're all a mix. You can't look at them and tell which one's which. Because you have both sensory and motor axons in the same nerve, they're called mixed nerves. Mixed meaning there's both sensory fibers and motor fibers in the same nerve. So all spinal nerves are mixed. Here's a cross section. The connective tissue is out here, bundling together, hundreds of axons. All of these little things here are cross sections of axons. A lot of this white is probably the myelin sheath, and then the material on the inside is the axon. So some of these are myelinated. You can see a myelin sheath around, that white tissue, or white stuff, not tissue, but white stuff around that axon is the myelin sheath. And in fact, the myelin sheath makes the nervous tissue more light in color, which in the brain we would call white matter, versus unmyelinated axons are darker and are going to be called what we call gray matter in the brain and spinal cord. So you also see there's blood vessels. There is a blood supply in the nerves. Oops. So hopefully you recall how nerve impulses travel to and from the spinal cord. Here's the nerve that we just saw. The lines here represent the direction of information, but it also represents axons. So we have the axons in the nerve. Sensory axons enter the spinal cord through the posterior nerve root. It's called the posterior nerve root. Remember the spinal cord, a very quick review, the anterior side, the front side has a deep groove in it called the anterior median fissure. That's an anatomical landmark telling you that's the front side. And then you have the gray matter, which looks like an H. The front ends of the gray matter are called the anterior gray horns. Anterior gray horns. The posterior side, the back side, you have posterior gray horns. You also have, in some parts of the spinal cord, you have lateral gray horns. And then you have the gray commissure, which connects the two sides, and the central canal, the cerebral spinal fluid in the middle there. The lighter colored areas are white matter. They're called the white columns. Anterior, lateral, and posterior white columns. They contain tracts, which are bundles of axons carrying information up and down, to and from the brain. Ascending or sensory tracts carrying sensory information up, motor tracts carrying information down, also called descending tracts. So this is the information highway right here. Gray matter, though, doesn't have much myelin in it, so it's unmyelinated. Anyway, so this is myelinated, unmyelinated. So sensory neurons, axons, are traveling down the nerve. They're going to enter on the posterior side through the posterior nerve root. Remember the cell body, this is a unipolar sensory neuron. Its cell body is in the posterior root ganglion. There is no ganglion on the anterior side. So that's another clue that you're looking at the posterior side. The axon enters, and here it's showing it may, the axon could just go on up through the tract, up to the brain, but here it's showing it synapsing with an interneuron. This represents a synapse between the synaptic end bulbs and the cell body and dendrites. This is multipolar neuron, this interneuron. And it's being stimulated by neurotransmitters from the sensory neuron at the synapse here. And if it gets stimulated enough, it'll generate action potentials down its axon. And so the axon runs from the back to the front. And so the cell body here is in the posterior gray horn, but the synapse here is in the anterior gray horn. It's synapsing with a motor neuron, shown in red. This is a somatic motor neuron. This would be multipolar, dendrites and cell body here. Axon exits out through the anterior nerve root and into the nerve, okay? So anytime you see a V in a circle, that is a synapse, represents a synapse. So you might have, and I'll show you a reflex arc. You have information going in and then information going out, always in that direction. But notice that you have both sensory and motor fibers in the same nerve. So the nerve itself is mixed, but the posterior nerve root is always sensory, the anterior nerve root is always motor. You also have, we're going to be talking about autonomic neurons soon. They're also multipolar, but their cell body and dendrites are located in the lateral gray horns here in the spinal cord. But the axon exits just like the somatic one does through the anterior root. And we're going to be talking more about autonomic neurons later. And so here this picture shows a reflex arc, which is, I don't know why the computers in this building have problems with, I'll have to put a different picture after this, because it's looking at a picture. For some reason, when I do it on my computer, it works, and school computers in the rooms don't. It must be the program they use. But anyway, you have here somebody getting, pricked by a needle or pin, or stepping on a nail, or you could be putting your hand on a hot stove, or a prickly cactus thorn, or something like that. You stimulate sensory receptors in your skin, pain receptors in your skin. They generate first depolarizing graded potentials, and then action potentials that get propagated down the axon. There's the cell body, there's the posterior root ganglion, this is the posterior root. The impulse travels into the posterior gray horn. This neuron stimulates an interneuron there in the central nervous system in the gray matter. And then what they're supposed to be showing you here is another neuron, and I'll have to just put in a regular picture, but anyway. And that motor neuron here would send out a signal out to an effector, which in this case would be skeletal muscle. So you prick your finger, pull away, or you step on a nail and you pull your foot up, or whatever, stimulation is. That's a reflex. It's a fast response to a painful stimulus, and it's a way of, protecting yourself. But I'll fix that picture and repost those notes. Any questions about that? That illustrates, though, how you have that flow of information in that order. And here you can also see it with this guy and the pressure receptor. Here you can see the sensory neuron entering through the posterior nerve root and the posterior root ganglion here. And then you can also see the motor information coming down from the brain, and the motor neuron, which we have an upper motor neuron synapsing with a lower motor neuron. That sends signals out to the anterior root and then into the nerve and to the effector organ. So you can see those structures in that picture as well. So this picture just highlights some of the functions of the spinal cord. And there's all in the book, in the textbook, the numbers correspond, in the text. So it describes each of these things. But let's say, I'm trying to see which one this one is. Okay, so this one might be a pressure receptor or a touch, well, it's probably a touch receptor, a Meissner's corpuscle, a touch receptor, stimulated by touch, sending signals through a unipolar sensory neuron, and the axon travels up to the brain through sensory or ascending tracts, which are located in the white matter, in the white columns, right? Sending information up to the brain so you feel that sensation. Another possibility is, in the second scenario, some kind of receptor in the skin gets stimulated, its axon travels in, same way, there's that posterior root ganglion and a bunch of cell bodies there, except that this sensory neuron synapses with an interneuron in the spinal cord, and that axon crosses over and travels to the opposite side. So in other words, if you feel something over here, that information is going up to the opposite side of the brain. By the way, this is going to cross over in the brain stem, in the medulla oblongata. So the information goes either up to the brain stem, up to the medulla oblongata and crosses over, or it crosses over in the spinal cord. Depends on where it's coming from. Another possibility is, I think this is the reflex arc. You prick your finger or burn your hand, and then you do a reflex, and you stimulate the muscle to contract and pull your hand away from the stimulus, okay? It also shows motor impulses coming down through descending motor tracts, and notice it crossing over. So motor information also, the axons cross over either in the spinal cord or in the brainstem, in the medulla oblongata. Both sensory and motor tracts do cross over in the brainstem, but some of these axons cross over in the spinal cord, and now it's stimulating this is, by the way, an upper motor neuron. Here's the lower motor neuron, somatic motor neuron, and now it's sending signals out to cause muscle to contract. But this would not be a reflex. This would be more like stimulating your hand to move, your arm to move, or something. So, the left side of my brain is going to stimulate the right side of my body, those muscles to move. And then finally, what we're going to be coming to soon, just a little bit, is the green one. This is an autonomic motor neuron. It's receiving signals from the brain as well. And it is sending signals out to its axon to a second neuron. So in the somatic system, the motor neuron, there's only one axon, one neuron that stimulates the muscle. In the autonomic system, there is what we call a preganglionic neuron, and it synapses with a postganglionic neuron within a ganglion, like this one, but further out, further away from the spinal cord a little bit. Autonomic motor ganglion, where there's a synapse between the first neuron and the second one. These are both motor neurons. And this postganglionic neuron can either stimulate the heart, or it could inhibit the heart, depends, but it could either stimulate or inhibit the heart. It could stimulate or inhibit urinary or smooth muscle, stimulate or inhibit secretions from glands. Now, I say stimulate or inhibit. It depends on if you're looking at the autonomic, or if you're looking at the parasympathetic or sympathetic division, which is what we're going to be talking about shortly. But It's not gonna do all three. It kind of makes it look like one neuron can do all three jobs. It can't. A postganglionic neuron is either going to synapse with cardiac muscle, or it's going to synapse with smooth muscle somewhere in the body, or it's going to synapse with some gland somewhere in the body, but it's not going to do all three. It's just kind of symbolically showing that. But this is the autonomic system, which is what we're going to be talking about next after we finish this up. Any questions about this picture here? Okay. Let's see, I'm not sure why I have this up there, but anyway, I was just going to say too, we were talking about somatic and autonomic motor systems and motor neurons. I do want to, or neurons, nerves, I should say, in general. There's also the cranial nerves, and the cranial nerves are cranial nerves 1 through 12, and there's pairs of them coming out from each side of the brain, and they're numbered 1 through 12 by Roman numeral, but they're also got names: olfactory, optic, oculomotor, and so forth. And so there's a mnemonic device for remembering them from anterior to posterior side of the brain. This is an inferior view of the brain. O, to touch and feel. Very good, velvet. You may have used that to learn those nerves. But they vary a lot in what they do and whether they're sensory, motor, or mixed nerves. Spinal nerves are all mixed. The cranial nerves are not. So I did just kind of a summary that you can refer to of the cranial nerves. And you can see that, like, for example, the olfactory, optic, and vestibulocochlear, number 8, are just sensory. They just do sensory function. We will talk about them a bit in the sensory system. Some neurons, though, are primarily motor neurons, like oculomotor. Oculomotor, like ocular is the eye. One of the things it does is controls eye movements. Trochlear is a motor nerve. It also controls eye movements of the eyeballs. Other nerves, though, are mixed nerves, where they have both sensory and motor function. So it just varies with the nerve, and it's just kind of a very general description of their functions that what they either sense or what they control. And really, it's the brain that's sensing and controlling, but they transmit that information from sensory receptors to the brain, or from the brain to the effector organs. Now, many of these nerves also have some autonomic functions, and they're part of the parasympathetic division that we'll be talking about shortly. So, for example, the facial nerve not only controls facial expressions, as the name implies, but it also controls some autonomic functions. It stimulates salivary glands to secrete saliva, and it stimulates tear production. Let's see, another one, let me see, the vagus nerve is a major autonomic nerve. It's probably the most important of all of them. I mean, they're all important, but this has the broadest range of functions because it controls all sorts of stuff, including digestive functions, respiratory functions, and the strength and rate of your heartbeat. It actually slows down your heart rate. It doesn't speed it up, it slows it down. So it stimulates digestion, but it slows down your heart. It's part of the parasympathetic division. So any of these nerves that have autonomic function, they're parasympathetic division nerves, as we'll see. But anyway, those are the cranial nerves, and we'll stop with, I guess, at that point. Any questions? Now, I'm not going to be testing you on all these nerves, but any ones that we do talk about in our discussions of the autonomic system. We're not done yet. Don't start putting your books away. We're just going to the next thing. See people starting to put stuff away. It's not time. But any nerves I do talk about in the future, whether I'm talking about in terms of the parasympathetic division, or whether I talk about sensory section or any of that, you do need to know those nerves, because they are in the context of those notes that we'll be talking about. You don't have to memorize all of this, but you do need to know the stuff that we deal with later on. But what I want to do is go on to, at least get started with the autonomic system. Those of you who've had me in class know I don't let you out early, so don't expect to get out 15 minutes early. That's just not going to happen. We don't have time to get out early. We've got to cover a lot of material in this class, so we've got to get it done. But anyway, autonomic system is what we're going to look at next. Some of you have had it, some of you have not. And if you have had it, probably wouldn't hurt you to see it again because it's, a pretty complex system. So, if you did see it, you probably saw at the end of the semester when you were rushing, trying to finish up and trying to get your final study for, so it's probably a good idea to see it again. Anyway, autonomic nervous system is the involuntary subconscious part of your peripheral nervous system. The central nervous system, of course, controls it, specifically the hypothalamus of the brain and also parts of the brainstem are involved in controlling autonomic nervous system function. So as a reminder, in the brain, hypothalamus is a tiny area. This is by the way, a mid-sagittal section. This is the cerebrum. That's where you're doing conscious thought. This is what controls somatic functions. But other parts of your brain, the cerebellum, the brainstem, the diencephalon, where the hypothalamus is, these parts of the brain control involuntary functions. And in the case of cerebellum, it is basically coordinating voluntary functions. It's coordinating your skeletal muscle contractions, but it's doing so at a subconscious level. But the hypothalamus right here, this region, which is just below the thalamus, in what's called the diencephalon region. It's a tiny area. It's also attached to the pituitary gland, which you don't see here, but the pituitary gland would be hanging off here. And it's a tiny area, but it controls autonomic functions. So it's a very important area you can't live without. If you damage the hypothalamus, then you can't control your involuntary functions of your body. Now the brainstem is also involved. This is the brainstem. You've got the midbrain, the pons, which looks like a little pooched out tummy or something, and then the medulla oblongata. Below that, when you get to the foramen magnum would be right here, at the base of the brain, at the base of, below the cerebellum. Then below that would be the spinal cord. So, the autonomic system is here in the middle part here, controlled again by the brainstem and hypothalamus of the brain. You've got both sensory and motor function. Sensory receptors, we're just going to talk briefly about them. They're also called interoceptors, which means internal receptors. Sometimes they're called visceral receptors, at least if they're in the viscera. And these sensory neurons and sensory receptors are involved in detecting changes going on inside the body, most of which you're not aware of. So these are changes going on in the bloodstream, chemical changes that happen in the bloodstream because you're transporting gases like carbon dioxide and oxygen, but you're also, carbon dioxide can actually make it more acidic. So you've got CO2 and oxygen receptors, you've got pH receptors detecting these chemical changes. You also, there's also blood pressure, that you can detect with these receptors, and other things. Also stretching, like stretching of the bladder or stretching of the stomach. Most of the time you're not aware of these sensations because it's at the subconscious level, but sometimes you do feel, if you feel pain, that's going to be from these interoceptors. If you feel nausea, if you feel stretching, like if your bladder's full, you are consciously aware of that. But most of those internal feelings you don't notice, you don't feel. Now, there's a couple different kinds of interoceptors. As I mentioned, chemoreceptors. These are ones that detect things like pH of your blood, because it's carbon dioxide. If you get too much carbon dioxide in your blood, it can cause your pH to go down, because CO2 and water in the blood form carbonic acid. So you can actually get an acid, which means that you want to get rid of CO2 from the bloodstream. You can measure, blood CO2 levels as well by chemoreceptors. There's also mechanoreceptors, which detect mechanical changes, and that would be things like stretching of organs, particularly organs like your digestive organs, your stomach and intestines, especially the rectum, and also stretching of things like the bladder, urinary bladder, and also the heart has stretch receptors in it. So if more blood is going into the heart's going to stretch a bit more. There's stretch receptors monitoring that. Those are all mechanoreceptors. And by the way, blood pressure. There's blood pressure receptors in your heart and in your carotid arteries, which are the arteries in your neck. You can feel your heart rate in your neck. You know, you can take your heart rate, but you can't feel your blood pressure. You can't sense it. That's why they often call high blood pressure the silent killer, because you can't feel it when you have high blood pressure. It's not something you're consciously aware of. That's why you should always monitor your blood pressure and make sure it isn't too high. Okay, now any questions so far? Now we're going to focus on the motor functions, because that's really when we talk about parasympathetic and sympathetic divisions, that's what we're talking about, are motor functions. This table can be very helpful because it summarizes some of the differences between the somatic system on the left in yellow and the autonomic system on the right. Somatic system is, deals with somatic senses, which means what you're consciously aware of, and special senses, which are what you see, hear, taste and touch, and also equilibrium. That's special senses. The control of motor output is control of voluntary control from the cerebral cortex, and also with contributions from basal ganglia and cerebellum, because they're coordinating those contractions, and brainstem and spinal cord are involved too, but that's voluntary control. There's a one neuron pathway. Remember, there's only one somatic motor neuron sending signals out to the muscle. Just one neuron. So if that neuron's axon exits here and runs down your foot, that axon is that long. It runs all the way down to your foot through the nerve. And all of the somatic motor neurons use ACH, which stands for acetylcholine. The effector is always skeletal muscle, nothing else. And it stimulates contraction of skeletal muscle. And so very simple. Autonomic is much more complex, as you can see. The sensory input is from interoceptors, though there is some input from your somatic senses, because your somatic system and autonomic systems are tied together somewhat, so that what you sense and what you feel and your emotions can be tied into your autonomic system, which is why when you're upset, you get an upset stomach, or you get, your heart rate goes up, your blood pressure goes up. Your emotions are tied into your autonomic system. So they're not separate. There is some communication going on, which one can affect the other. Motor function, involuntary control primarily from the hypothalamus and brainstem. Limbic system, I don't know how many of you have studied the limbic system, but that is a system that's involved in emotion. So your limbic system, your emotions, in other words, can have an effect on your autonomic system. And then maybe some limited effects from the cerebral cortex, particularly if you're looking at something like urination, and going to the bathroom. You can have conscious control. You don't just go to the bathroom whenever. You do control your bladder, you control whether you defecate, so there is some cases where you have to have cerebral cortex control, but it's mostly involuntary, but a little bit of voluntary control in those cases. Instead of a one neuron pathway, you got a two neuron pathway, as I pointed out earlier. Preganglionic neuron synapses with a postganglionic neuron that directly stimulates. It's the postganglionic neuron that stimulates the effectors. And that's in most cases. There is one place where there's not two, there's only one, that's with the adrenal cortex. We'll talk about that later. Neurotransmitters, it gets much more complex. We'll talk about that a little bit later, but there's more than one neurotransmitter. There's acetylcholine. There's also what's called norepinephrine. So there's a couple of different neurotransmitters. They have different effects. Effectors are smooth muscle, cardiac muscle, and glands, and the responses can be either stimulatory or inhibitory, either stimulating contraction or inhibiting muscle contractions. It could be, stimulating or inhibiting cardiac muscle and heart rate, stimulating or inhibiting glandular secretions. It depends on which type of neuron is stimulating those effectors, as we'll see. So this is a good summary to look at to help you understand the differences and the functions of the autonomic system. Most of your internal organs, and that includes blood vessels and heart, as well as your digestive and urinary organs and respiratory system. And it also includes glands, both exocrine and endocrine. Most of your internal organs, except for skeletal muscle and bones, you could say they're not really affected, I don't think, by the autonomic system, but most internal organs have dual motor innervation. In other words, they are innervated by both the sympathetic division and parasympathetic division, which have opposite effects on those effectors. Parasympathetic division, sympathetic. This is the motor portion of the autonomic system, not the sensory portion, just the motor. These two systems have opposite, or what we call antagonistic, effects. And they are activated and respond depending on what's going on in a person's life at the moment. Are you resting and digesting a meal, or are you going for a run, or dealing with an emergency? Those kinds of things. There's different times require different reactions. The sympathetic division is what stimulates the fight or flight response, or what's called fight, flight, and stress response, but you can also include exercise in this. When you are fighting or fleeing a situation, an emergency, you're being chased by a bear and you're either going to fight or flee, from that bear, your sympathetic division is going to be kicked in big time. Or you hear about people who lift heavy objects off a person or push a car off a person, or things like that. That is mediated in part by the sympathetic division. You might think, well, what about adrenaline? Well, that is part of it too, because the sympathetic division stimulates the adrenal medulla, the adrenal gland, to produce adrenaline, which also helps facilitate the fight or flight response, particularly if there's a big emergency or something like that. But the sympathetic division also is active when you're stressed out, both physically or emotionally stressed, and it kicks in when you exercise. So it's also involved in activating the body when you're exercising. Parasympathetic division, on the other hand, mediates what we call rest and digest activity. So at rest, that's the division that's most active. It's also called the feed and breathe system, because parasympathetic division not only stimulates things like digestion, urinary output, it also facilitates certain reproductive activities, especially during sex. So it's very active during that time. So feeding and breathing, or resting and digesting, all of that is controlled by parasympathetic, so it conserves energy, it stimulates digestion and urinary output, it slows down your heart rate, slows down the respiratory rate. On the other hand, sympathetic division, the fight or flight response, increases your alertness, it allows you to act quickly, it increases your heart rate and breathing rate, it dilates your pupils, it sends more blood to your muscles, it also slows down digestion and urinary output. It also stimulates sweating, so you sweat more when that sympathetic division is kicked in. We'll talk more about these on Tuesday.